Welcome. In this video, we are going to consider receptors. We're looking at principles of drug actions, targets of drug actions. But to give us a broad and general idea, we are supposed to have some basic concept pertaining receptors. So let's move into that. Now, what are receptors? We say receptors are macromolecules or complexes of macromolecules that have two functional domains. So every receptor should have two functional domains. Initially, we were trying to define receptors as proteins, but we realized that DNA, some anti-cancer drugs, binds to DNA and cause pathological activity or intrinsic activity. I mean that DNA is not a protein, but it is a receptor. So if I'm trying to define receptors proteins, that means that I'm taking DNA out. So we change the definition from proteins, and now we say that receptors are macromolecules or complexes of macromolecules that have two functional domains. And this functional domain, chemical substances bind to and causes biological action in a biological system. So we are saying that to call something a receptor, it should have two functional domains. Now, what are these two functional domains? These are the two functional domains. So to call something a receptor, the receptor should have a ligand binding domain. And the receptor should also have an effector domain. Anything that has a ligand binding domain but do not have an effector domain is not a receptor. That is why plasma proteins are not receptors. Why? Because they are having a ligand binding domain, but they have no effector domain. So drugs can bind to them, but they do not cause any intrinsic activity. So plasma proteins are not receptors even though drugs bind to them. Now the effector domain part of the receptor is going to cause this kind of intrinsic activity that we're looking for. So a receptor should have a ligand binding domain, where that means it should have a part where a ligand will bind to. Now, what is a ligand? A ligand is any substance that binds to a receptor. So this ligand is binding to a ligand bind domain of this receptor. Ligand is anything that binds a receptor. So a ligand can be a drug. Neurotransmitters are ligands. Hormones are ligands. Drugs, as I've said, are ligands. Even toxic substances that bind to receptors are ligands. So anything binding to a receptor is called a ligand. And they bind to the ligand binding domain of the receptor. And if they should bind to the ligand binding domain, they should stimulate the effector domain to cause some kind of intrinsic activity needed. This is a receptor. We have two functional domains: ligand binding domain, effector domain. Right. And now we know ligand, and we know examples of ligands. Now let's go to the distribution of receptors on different parts of the cell. We must be knowing that most students think that receptors are found only on cell membrane. So let's suppose this is a cell. This is a cell membrane. So most students think that receptors are mostly found on the cell membranes. That is no. So one, I can see receptors on the surface of membranes. So I can see receptors here the surface of membranes, that is area 1. I can also see receptors in the cytosol, that is area 2. I can see receptors inside the cell, the cytosol. Receptors can also be in the nucleus, area number 3. And receptors can be outside the cell, area number 4. So we must be knowing that when we talk about receptors, we can see some of them on the surface of cell membranes. That is area number 1. We can also see some of the receptors in the cytosol, in the cell, area number 2. We can also see receptors in the nucleus, area number 3. And some of the cells, we can even see them outside the cell. That becomes area number 4. Now let's go to different receptors. Now, starting off with this one. This is a transmembrane receptor. Okay, this is a transmembrane. Okay, let me put it to transmembrane TMR. Transmembrane receptor. Of course, it's having the ligand bind domain and the effector domain. So this is really a receptor. And this is the transmembrane part of the receptor. So for this receptor to be activated, a ligand should come bind to this receptor and to activate the effector domain, and the intrinsic activity will be initiated. So this is a transmembrane receptor. The next type of receptor I talk about is ion channels. Ion channels are 4 to 5 pass receptors. This is an ion channel. It is a 4 to 5 pass receptor and found on cell membrane, similar to the transmembrane receptor here. Now, this is an ion channel. It is having a ligand bind domain, meaning a ligand should bind to this. So when a ligand binds to this ligand bind domain of the receptor, then the channel will open, and therefore ions will be entering. Ions will be entering, and the ions are going to cause that kind of intrinsic activity needed. So the ligand must first come, bind to the ligand bind domain, and when it binds, it will cause the channel to open, and then there will be ion influx. That is called the intrinsic activity. Of course, this is also having a ligand bind domain, but this is having a special form of effector domain. That is the channel opening. So here there is no channel opening. So here when it binds, it's stimulating. But for this one, it is having effector domain, and that is the channel inside that the ions will pass through and come into the cell and activate the cell. That is ion channel. And they are 4 to 5 pass receptor because they span the membrane 4 to 5 times. The next receptor I talk about is 7 pass receptor. I also call it gene protein coupled receptor or serpentine receptor. Now, by all these names, it is called a gene protein coupled receptor because this receptor is coupled. It works by binding to a gene protein. Okay, so this is a gene protein. So this receptor is coupled with a gene protein. That's what is called a gene protein coupled receptor. It is also called a serpentine receptor because it looks like a snake. Serpent. So it is also called a serpentine receptor. It is also called 7 pass receptor because it spans the membrane 7 times. 1, 2, 3, 4, 5, 7 times. So it's also called 7 pass receptor. Right. Now, another set of receptors. So here, a ligand will bind the ligand bind domain, and the effector domain, okay, will activate the gene protein, and then the necessary cellular effects will be observed. Right. Now, the next set of receptors to talk about is 12 pass receptor. And it's called a 12 pass receptor because it spans the membrane 12 times. And a critical example is at level of the presynaptic neuron. okay, where norepinephrine will bind to, and it auto inhibits the release of norepinephrine. So it is mostly found on the presynaptic nerve terminal, all right, or membrane. So these are the kinds of receptors. So if nothing at all, know this transmembrane receptor, and this is the transmembrane part of it. It's having a ligand domain, having an effector domain. So it qualifies to be a receptor. This is an ion channel or 4 to 5 pass receptor because it spans the membrane 4 to 5 times. It is called an ion channel because a ligand will come and bind to the ligand bind domain of the receptor, and the channel will open for the ions to come inside. So the ions opening is the effector part of the receptor, so it still qualifies to be a receptor. So that here the ligand will not activate. The ligand will only bind and open the channel for ions to come inside. But without the ligand, ion channels will not open. This is also a 7-pass receptor because ligand binds here and activates the effector domain here and stimulates the gene protein to cause necessary activity. Off-pass receptor, ligands bind, okay, ligand will bind, and to activate the ligand, to activate the effector domain, leads to intrinsic activity. What have I said so far? Receptors are macromolecules, okay, that should have at least 2 domains, a ligand bind domain and effector domain. Ligand bind domain are parts where a ligand should bind to. Okay? And a ligand is anything that binds to a receptor. So a ligand can be a drug, a ligand can be a neurotransmitter, a ligand can be hormones, a ligand can be toxic substances, and many others. When it binds to that, then it should stimulate the effector domain to cause intrinsic activity. Now, we know that receptors are not found on the surface of cell membranes. Now, we know that some of them are found on the surface of cell membranes. Some too can be seen in the cytosol, some too are seen in the nucleus, and some too are even found outside the cell. Now, initially, so one thing is that ligand binding to receptor site, a ligand binding to the ligand bind domain are called first messengers. Giving them name, they are going to start everything. So they are the first messengers. First messengers. So ligands are first messengers. Okay? They're the signaling molecule. So they are the first messengers. So put in mind, ligands, first messengers. Now, initially, they were thinking that when ligand bind, let's put this for example. Now, when ligand bind to a receptor, they were thinking that it directly causes it arise. Fine. When a ligand binds to a receptor and activates the effector domain part of the receptor and activates the gene protein directly stimulates cyclic AMP. So they were saying that then cyclic AMP is a second messenger. So ligand or the signaling molecule is the first messenger, and cyclic AMP is the second messenger. Later on, they find out that no, there are some intermediaries that goes on. So for instance, if this is stimulated, the gene protein does not directly stimulate cyclic AMP. No, but there's some kind of enzymes over there that will get stimulated first for cyclic AMP to be activated. So now let's dive deep and know how the actions of this comes about. For instance, I've taken a drug. I want to get healed. When it binds to this one, what are the things that's supposed to go on here for me to get my cellular effects? But here it's simple. Ions must come inside. And when ions come, it causes that kind of intrinsic activity. Okay, and I get healed. But here, when the ligand binds here, drug binds here, what are the steps go through? This is the biochemistry level. That will go through for me to get a cellular effect that I'm looking at. So let's look at that. Right. So this is the serpentine receptor or G protein coupled receptor or anyone? G protein coupled receptor, serpentine receptor, 7-pass receptor. How am I calling this a receptor? Why? Because it's having what? a ligand bind domain that a ligand will bind to, and it's having the effector domain here. Okay, having the effector domain. Now, so a ligand is a drug should get bind here. Now, this is a gene protein. And when I talk about gene proteins, we have monomeric gene proteins and we have trimeric gene protein. So the gene protein that is binding with the receptor here is a trimeric gene protein having 3 subunits, alpha, beta, and gamma. Okay, good. Now, what happens if I want to get healed? If I want to get that kind of cellular effect, then calcium must be released into the cytosol of the cell. Let's say this is a cell. This is the cytosol. This is an organelle called endoplasmic reticulum. Now, if I want to get the cellular effects, I will have more of calcium rather in the cytosol. But here lies the case, the storehouse of calcium is the endoplasmic reticulum. So I must release some of the calcium from the endoplasmic reticulum into the cell. And when that happens, I am going to get that kind of intrinsic activity, that effect that I'm looking for, the drug. So how can calcium get into the cytosol? That's why you're looking at the various steps. So one, the drug that I will take, the ligand, should now come and bind to the ligand and domain. It should activate it. When it activates, it will then activate the effector domain. You know what? The effector domain is going to stimulate the gene protein. Okay, the gene protein. Now, the inactive state, without the ligand, now without the ligand, this one is not active. It is having the alpha subunit is having GDP, and that makes the gene protein inactive. So it becomes active when a ligand binds to it. So the moment a ligand binds to this one, alpha will detach from the beta and gamma subunits, and this GDP will be converted to GTP. Here, the alpha subunit has detached from the rest of the subunits. Why? Because a ligand was able to bind to a ligand binding domain, and it activated the effector domain. So it's rather activated the G protein, and then it has converted GDP of the alpha subunit to GTP. So this is the active form, okay, of the gene protein coupled receptor of the G. I'm sorry, of the G protein active form. But here, no, that GQ. We also have subtypes of gene protein. We have GQ, we have gene S and gene I. Gene stimulatory the gene S and gene I the inhibitory. So if the gene protein is inhibitory, that means that it's going to inhibit cellular effects. But the gene Q acts as the gene stimulatory. They're all stimulatory, but they act differently. So here, the alpha subunit is activated and now converted from GDP to GTP. And because of that, it has detached from the beta and gamma subunits. So now I have there my alpha GTP. Don't forget that I want a cellular effect where calcium should come to the cytosol. And when that happens, the cell will be happy. Okay? So they call the cellular effect. The effect that I want from the drug is happening. Why? Because calcium has been released from the endoplasmic reticulum to the cytosol. So how does that come about? So the alpha GTP that has been activated will now activate a special enzyme called phospholipase C. Now, when phospholipase C gets activated, it will go to the lipids on cell membrane and break them down into IP3 and DAG, dihydroglycerol, and inositol triphosphate. And this IP3 is needed to come and bind to the calcium channel on the endoplasmic reticulum. Now, when it comes here, release the phosphate group, comes here and bind, it opens a calcium channel. And therefore, calcium from the storehouse of the endoplasmic reticulum will come out from the calcium channel and come into the cytosol. And therefore, I've got my cellular effect. Now that the effect I want from the drug has been established, but calcium has been released into the cytosol. One more time. Ligand binds. When ligand binds, it activates the effector domain. And when it activates, this is the inactive state of the gene protein, and it's JQ. So JQ activates phospholipase C, but GS will activate adenylyl cyclase. So note, so the ligand binds here, okay, and activates the effector domain. This is the inactive state, carbon alpha GDP. And the effector domain stimulates the G protein, the GDP will be converted to GTP, and the alpha will detach from the beta and subunits. So now I have here my alpha GTP. Alpha GTP is going to stimulate phospholipase C. Phospholipase C is going to lipid on cell membrane, break them down into IP3 and acetyl triphosphate and DAG, dihydroglycerol. This is also going to cause phosphorylation. Now the IP3, okay, is coming to the calcium channel on the endoplasmic reticulum because that's having the calcium. And we need to bring it out to the cytosol. So when the IP3 binds with it opens the calcium channel for more calcium to leave and come into the cytosol. Therefore, aborting my cellular effect that I'm looking at. So this one is GQ. So GQ stimulates phospholipase C through alpha GTP. Let's go to inhibitory. Inhibitory. If this gene protein is an inhibitory one, it is going to inhibit. You understand? But here it works in a different pattern. So let's talk about the stimulative part first, then I'll come back. So this one is the gene S. So that one, the GTP will not activate phospholipase C, but to activate different enzyme called adenylyl cyclase. So let's say I have my ligand binding here. Okay. I have my ligand binding here. Oh, sorry. I have my ligand binding here. So to activate the receptor and activate the effector domain, and when it activates this, for this gene is gene stimulator, it's going to stimulate. This effector domain is going to stimulate the gene protein, okay? And here in the inactive form, without the ligand, without the drug, without the ligand, it is alpha GDP. But the moment drug comes here and this one gets stimulated, it stimulates the alpha and therefore convert the GDP to GTP. So here the activator form of this one is alpha GTP, and the alpha will detach the GTP from the beta and gamma subunits. And I have here my alpha GTP. Alpha GTP is going to stimulate adenylyl cyclase enzyme, and this enzyme is going to convert ATP to cyclic AMP high levels. And when that happens, more cyclic AMP is going to activate protein kinase A, an enzyme. And the kinases, what they do is that they phosphorylate. When they add phosphorylate group to the calcium channel of the endoplasmic reticulum, it is going to bring out more calcium to the cell, and I'll get my cellular effect. So phosphorylation binds to the calcium channel, it opens the channel, and therefore the calcium that is in the endoplasmic reticulum that needs to be in the cytosol to get my cellular effect when I come out and come to the cell, and I'll get my cellular effect. So ligand binds here, it activates alpha GDP out alpha GTP will stimulate to alpha GTP. And the alpha GTP will stimulate adenylyl cyclase. Adenylyl cyclase will stimulate adenylyl cyclase will convert ATP in the cell to more levels of cyclic AMP. And cyclic AMP also activates protein kinase A. And I'm saying that for kinases, they add phosphate group. So if phosphate is added to calcium channel, it is going to open a channel. So here, it comes here and binds the channel, also groups the channel, and therefore calcium will leave endo to the cell. Now get my cellular effect. That is how it happens. The same thing here. That is gene inhibitory. So you have different types of the gene proteins. Gene inhibitory, it is going to inhibit. So when a ligand binds here, since this is gene inhibitory, this gene protein is inhibitory. When a ligand binds here, okay, now it cannot activate this one. It cannot activate the alpha, it cannot activate the G protein to go and stimulate adenylate cyclase. Okay, it's not going to stimulate adenylate cyclase. So here, even though the alpha UTP will not be detached from here, so this thing will not even form because this will not be, even though there's a ligand, but because this G protein is inhibited, there will not be active form of the gene protein here, and therefore alpha GPP will not be formed. And when it is not formed, adenylate cyclase will not be activated. And when they're not activated, they cannot convert ATP to cyclic AMP. And when I have little or no cyclic AMP, I have little or no stimulation of protein kinase A. And therefore, I have little or no phosphorylation. And therefore, there will be no calcium leaving the endoplasmic column. And therefore, I will not get the cellular effect that I need. This is all about receptors and about gene proteins, couple receptors you should know. Now let's go to the main slide and talk about targets of drug actions. Thank you. But you must be knowing that there's gene S, gene Q, gene I. Gene I is going to inhibit. Gene Q will stimulate phospholipase C. Gene S will stimulate adenylate cyclase. In all of them, there's a release of phosphate. Thank you.